continue to grow and see many amazing early career members joining this fantastic metabolomics movement. The topic of discussion for this session is precision medicine and proactive health. And, precision health and precision medicine, we are going to talk about optimization of taking care of someone who is sick, someone who is taking a pill or a medication. Can metabolomics add information to help us assign the right drug for the right patient at the right time? A topic that we have been working on for the past 20 years. And then the proactive health. Can metabolomics inform us about where we are in terms of health? Are we in a healthy state? Are we dropping off from a healthy state to a disease state? Hopefully not. And can we slow this process of going downhill towards a disease? Can we stay in an optimal metabolic health and avoid transitioning into a disease state? In either case, we want to make health better, and together we are talking about precision approaches in our health. For the agenda of today, we have several speakers lined up. David is going to talk more about this concept of precision health and proactive health and the use of AI and an N of 1, technologies and AI tools that can enable us achieve what we claim we can achieve. Susan Sumner is going to talk about the all of us, precision medicine, nutrition initiative, and can we optimize the right diet for an individual, another big important topic. Matej will talk about early life, how early life can impact things down in the future. Important to think about health as a continuum that starts very early on. What happens early on could impact what we see later in life. Tuulia will talk about biomarkers, molecular insights. If we see things going downhill, what does it mean? Can we understand molecular mechanisms? And then Jennifer will talk about translation. Of course, we want to be able to translate all molecular insights into something actionable in the clinic. Just quickly, as a metabolomics community and over 2 decades, right, we have been making the statement that our health is impacted, and I don't have a pointer. I don't know how we point, but what you see on the left are things that influence our health. Of course, we know that the genome is a major contributor to changes in health, but that is not all. We know that the exposome, things we are exposed to from the environment, the water we drink, and so on, has a big influence. The diet, my goodness, the diet and what we eat, and these millions of compounds we ingest could have a profound effect on our health. Sleeps, drugs, supplements we take, all of this, and socioeconomic factors and disparities could impact our health in profound ways, and we see the metabolome at the center. This metabolome, the measure of thousands or maybe in the future hundreds of thousands of chemicals, are a great output for the net influences of all of this on our health. It's dynamic, it's changes, and we see that this, along with digital recordings that we get from our watches, right? Many of us are health aware. They are monitoring health. They are measuring blood pressure, glucose, EKGs, and so on. So the digital and the molecular coming together, this is where the dynamic data is. This is data and change in health in real time. And when we talk about proactive health, we want to be able to monitor this change and not go to a physician once a year to tell me, here is your glucose or here is your blood pressure. I'm monitoring it every single day, every minute. I have a recording on my watch. I'm getting my metabolic profile, so I'm learning a lot of information about health in real time. The genome is static, the metabolome is dynamic, and while we initially struggled, why is the metabolome so dynamic? Can we capture a metabolic signature? As it turns out, the metabolome being dynamic is a fantastic opportunity to learn about health in real time. So the output of all of this is changes in our metabolism that we are tracking, and this is what I just talked about. As a community, and we might call ourselves different things, metabolomics, lipidomics, exposomics, foodomics, end of the day, we are truly measuring a large number of small molecules that captures the net influence on our health. So I really want to make sure we do not end up falling in a trap that says, here is metabolomics, here's lipidomics, and here's exposomics. We are all one community measuring, small things in different tissues, different organs. The ultimate goal is to look at each individual, what is changing in an N of 1, how is my health changing in real time, and who looks like me is another important topic. I want to be able to relate and talk to people who share a common features. I'm going to make very quick remarks on 5 consortia we have led, 6 by now, 6 NIH-funded consortia in metabolomics. We were lucky, we were ready, we were, so money flew into enabling us to study the metabolome as part of precision health and precision medicine. These different consortia, including study of Alzheimer's disease, the exposome, pharmacometabolomics, have all been fantastic, enabling communities come together, learn together, putting the best minds around the table to learn about health and disease. Here is, for example, a snippet of some of the people we work with and collaborate and have collaborated with in different initiatives. I want to talk about precision medicine. Taking medicine and optimizing which drug should I be on. I'm worried about becoming diabetic. Which drug, what should I be on is something that really we understand. Diseases are heterogeneous. There's a lot of variability within each disease, and there's variability in how a drug impacts us and impacts our metabolism, based on a whole array of things, including gut microbiome and diet and lifestyle and much more. Therefore, we took upon ourselves and we partnered. We got a seat around the table in quantitative systems pharmacology, NIGMS, wanted to bring big data so that pharmacology can become more quantitative and far better informed. Where they started with the genetics, we added the metabolomics, and we said, listen, instead of just pharmacology being waiting for a clinical endpoint, we wanted to study real changes in real time as we take medications, and we partnered in the study of 10 classes of therapies commonly used, antidepressants, statins, antihypertensives, and on and on and on. By using metabolomics in thousands of individuals, yes, we were able to learn a lot of things. We started with the study of antidepressants. We realized quickly, if we profile at baseline, we can see that people at the get-go, the people who end up responding very well, the people who do not respond at all and are resistant, if you profile them metabolically at baseline, they almost have a different flavor of disease. They arrived at the clinical symptoms by activating different pathways. We mapped the influences of 3 SSRIs, rapid-acting ketamine, placebo effects, and much more, creating a mega database on response to depression. And the results were astounding, telling us really metabolomics can inform a lot. And if you add metabolomics and genetics, my goodness, it becomes even more powerful. A concept we developed with the Mayo Clinic, pharmacometabolomics informed pharmacogenetics. Big data and some data, some of this biochemical data can tell you exactly who might respond, who might not. We looked at statins. My goodness, statins, a difference between day and night. I cannot point on the left. People who respond and do not respond have different lipidomics profile. People on aspirin have a whole array of changes, variations between men and women. People on antihypertensives, we saw major differences, of course, in response of African Americans and white, Caucasians, and we saw the metabolic imprint, that goes along with variation of response. These were some of the early findings, followed by many more studies that taught us metabolomics can inform about variation in response. The drugs, just to tell you that the gut microbiome, these are interconnected highways from the exposome, the gut microbiome, the diet, all can influence our metabolic states, and they are interacting. Much of the effects on the gut microbiome is really drugs are a big influencers on the gut microbiome, on the metabolome, and we have been able to use big data to rethink drug discovery and drug development. We don't have time to talk about this right now. I just want to exemplify 1 or 2 minutes, how are we studying disease and sustenance of health? While we think about drugs now, we want to think about disease and progression towards a disease. In the study of Alzheimer's disease, which has been a very big investment in metabolomics in our consortium, we have been able to create data on over 40,000 individuals from Alzheimer's studies, from longevity studies. I thank you. Here's a wonderful gesture. We have been partnering as a community, right, with over 120 scientists from over 40 academic institutions, studying aging, changes during aging, studying Alzheimer's disease, building atlases, databases. But the most important thing is that we are mapping these biochemical changes in the blood. We are connecting across the trajectory of the disease. We are looking 20 years before. What are these biochemical changes that happen in people who develop dementia and end up in trouble? We are adding the influence of the genome on the metabolome. We are connecting the influences of the gut microbiome, the diet, the socioeconomic factors, and connecting changes in the blood with changes in the brain and imaging. The lessons learned, we are redefining, using this biochemistry, we are redefining our understanding of brain metabolic health, and early changes and peripheral changes that lead ultimately to change in brain and cognitive function. We are adding molecular data on each individual from 10 Alzheimer's studies, where we are adding metabolomics, lipidomics, exposomics, foodomics, metagenomics, adding all of this towards a precision medicine approach to understand the trajectory within each individual. Who is transitioning, who has pathology in the brain but does not transition, and much more. Just to give you a live example that this is happening, and it's happening at a big scale, by bringing, again, great people together. We are stratifying the disease. We are looking into what are these changes in biochemistry that happen in people who are resilient to development Alzheimer's disease versus those who are really susceptible and end up in a big decline, pointing to mitochondrial energetics, pointing to specific molecular processes that can tell us who is converting, yes, and we are developing tools to be able to understand what is my mitochondrial capacity index. By we, this is our German partners, Matthias Arnold and Gabi Kastenmüller and Jan Kramsiek and others, that brought in data from 10,000 individuals, where we are defining now, using some SNPs and metabolites in a very targeted way, who is susceptible, what are these biochemical changes, and can I stop it, is the ultimate goal. This is a real example of proactive health, trying to put an individual in a metabolic state that is stable, stopping and hoping that the disease will not progress as fast. If we can use these approaches along with drugs that we take, we see that the combination of all of these approaches is the way for sustenance of health. All right, to do what we do, we are communities of people bringing together foodomics, exposomics, drugs, drug metabolites, lipidomics, a lot of things, and we are adding socioeconomic factors. We don't have time for all of this, but I also wanted to add just a word on something that Susan Sumner will comment on: diet. How do the diets impact us, and how do they impact us differently? We have done studies now with ketogenic diet, Mediterranean diet. The pointer is a Mediterranean plus adding a diet and lifestyle change. And we have done the Dean Ornish diet, and we are mapping and building databases on what these diets do to metabolism and how do they revert or change metabolic profiles to something more favorable for aging and preventing Alzheimer's disease. I don't want to tell you what we see as differences, big differences, for example, with the ketogenic diet, variations among individuals, and much more. I'm going to stop here. Just to tell you that this is all communities coming together. We have been building communities for 20 years. We always work on lifting up, bringing communities together. I hope we can learn something over the next few minutes, over the next hour, on how we as a community can get a seat around the table And we are working on data harmonization, bringing together metabolomics data from different platforms. We synergize with a prior workshop that took place just at 8:30. Fantastic effort in biorepositories and how do we make data more usable and more, used by a whole community. All of our data is public. I'm going to stop here and ask Dr. David Wishart to give his perspective on precision medicine and precision health. I need to.
Thanks very much, Rima. Good to see a big crowd today. So we want to make sure there's enough time for discussion, so I'll try and be relatively quick here. Make sure I can get this working. Okay. So I think when we talk about precision, we have to also think about things like accuracy and precision. So the formal definition of precision refers to the quality of being exact and consistent. That doesn't necessarily mean accurate. And I think when we talk about precision medicine or precision health, I think we ideally want to think of accuracy. But precision sounds better than accuracy, so I think that's probably why it was chosen. So if we have both high precision and high accuracy, we do really, really well. And I think that's the aspiration that we would have for precision health and precision medicine. Ideally, we want to know in whether it's precision health or precision medicine exactly where the problem is and exactly how to treat that. And the evolution of precision medicine through to precision health has been sort of a circuitous process. Precision medicine is an older field. It's been around longer. The term is used more widely. Precision health, I think, is more, it's newly emerging. As Rima highlighted, precision medicine really focused on using omics methods, so that's genomics, proteomics, metabolomics, transcriptomics, to give that precise diagnosis of diseases, but also giving you more precise treatment. In the end, people say it's really like trying to get the right drug at the right dose to the right patient at the right time. That concept is great and it works, but it still falls in this domain of what I call reactive medicine. You wait until the disease appears and then you try and treat it. That's the way that medicine has always been. And it's typically a case of you still go into the doctor, a whole bunch of mysterious tests are run, and then you get the verdict, and then you go in for more tests, and you get another verdict. So it's not a lot of patient engagement. The shift to precision health, I think, is this idea that there is active engagement, that people are involved with their health. It's more proactive. It's something that you're not simply waiting for a verdict. It falls in the domain, I'd say, of public health, which is something that only gets about 5% of the medical budget in most places. It still uses omics methods, but it also incorporates a lot of artificial intelligence. And the idea is to precisely and personally optimize health. It's to focus on disease prevention, enable early disease detection. And Lee Hood, I think, has sort of summed it up many times. He's called it predictive, preventative, personalized, and participatory. And I think that concept really sums up this idea of proactive health. So when we talk about precision health, we're trying to use, in some respects, AI to help with that early detection, things happening at the earliest stages. Most of us are in some level of reasonably optimal health, especially if you're younger. And then over time, things start going a little awry. And those changes take place over 20 or even 30 years. And there's good evidence for things like diabetes, heart disease, Alzheimer's. All of those seem to happen 20 to 30 years before symptoms actually manifest. Typically, it's in the late stages where medicine is typically picking these things up. And that's a problem because often diseases are more treatable if you get them in the earliest stages. And we usually use the example of cancer, but it's true for diabetes, it's true for Alzheimer's, it's true for Parkinson's. So whether it's precision health or precision medicine, the idea is using the omics methods to identify biomarkers. And in many cases, at least the focus for today is about metabolite biomarkers, but often you can use proteomic biomarkers as well, and I'll show some examples of that. So this is a picture I often show, and I'll show it again, but it's sort of this connection between the genome, the proteome, and the metabolome. The genome is largely unchanging. The genome codes for the proteome. That does change to responses to both environment, diet, microbes, and physiology. But the one that's most affected by both the environment and technically by the genome is the metabolome. It lies at the interface between phenotype and genome. And so that makes it a particularly useful tool for monitoring things like the physiological influence, anatomical changes. Many of the tissues in your body have very profoundly different metabolomes, but identical genomes. Microbes profoundly influence your metabolome, as does your diet, as does the environment. What we sometimes say is that the genome tells you what might happen. The proteome tells you what happened a while ago. Could be a few days, could be a few weeks or a few months. The metabolome tells you what is happening. The microbiome tells you what others, primarily the microbes, are doing to you. And so all of these things have an important role in precision health and in precision medicine. But again, I think for the people in this room, the metabolome seems to have the most significant influence. And this is what we're trying to do, as I said, for both precision medicine and precision health. Now, the concept of precision health has been emerging, and it's been more driven by, I think, consumer organizations or commercial organizations. So for a number of years, people have been able to measure their genome. You can do this through Ancestry, 23andMe, sort of, Nebula. So these are commercial tools that cost anywhere from $50 to $500. There's only one or two companies that actually measure your proteome, but you can get it done. There are also a small number, although growing, of companies that measure your metabolome. And some of these are in Canada, the US, and in Europe. Many more companies actually involved in measuring your microbiome. And to some extent, the advances in precision health have been driven by these companies. And at some level, academia has been trying to catch up. But it's also been evident that there are issues with these systems. Some are less scientifically supported, others more so. Others, it's challenging to collect samples. Others, the measurements are rather modest. So really, to make precision health perhaps more useful, more viable, there still needs to be some technology development. One of the key areas I think that's important is to have absolute quantification, because this allows you to do broad comparisons across different platforms, different organizations, but also to standard reference values. There are thousands of standard reference values. All of them are quantitative for various biomarkers in the body. Ideally, measuring just a few dozen compounds, as many commercial tests do, isn't sufficient. You really need to have hundreds, if not closer to 1,000, high-value biomarkers that cover both endogenous and exogenous compounds. Those would capture elements of both the diet, exposure, minerals, but also endogenous metabolites. One of the things that we've learned from the genomics world is that just spitting into a tube is much easier than drawing blood. And to do metabolomics, we have to draw blood. To do proteomics, we have to draw blood. Ideally, what we want to have or develop are painless, inexpensive systems to extract plasma or serum for at-home collection. And then to also permit a low-cost mail-in. So if anyone's ever done an Ancestry or 23andMe test, it's spit in a tube, mail it off. If anyone's had to do blood collection at home, you realize that you have to have a phlebotomist, you have to have centrifuges, you have to have refrigeration systems. And then in the end, when you produce a result, if you got it from 23andMe or Ancestry, it's easy to read, it's easy to understand. Ideally, if you're to do the same sort of thing with the metabolomics world, they also need to be easy to read and easy to understand. Within our own group, we've been actively working and developing low-cost, high-throughput, quantitative assays. We've also been working on the development of facile, painless plasma collection systems that don't require any mechanical separation. They just use simple passive filtration, and they use something called a TASO blood collection device to help separate the plasma. So by moving towards low-cost plasma collection, low-cost metabolomics testing, this would enable not just one-off genomics testing as we currently do, but continuous or regular metabolome monitoring or proteome monitoring. The other thing that's emerging is the availability of low-cost wearable technologies or cell phone technologies to also support continuous assessment, continuous monitoring. This gives you information about phenotypic results, blood pressure, heart rate, or clinical components with blood glucose or breath pH monitoring. If you can integrate these continuous omics measurements with continuous phenotypic and clinical monitoring, then you can start doing this N of 1 experiment, doing it on you or with your doctor or your dietitian. And by doing that continuous monitoring, now you can start employing tools like AI to interpret all of this, because there's a lot of input. In the end, it allows you to do personalized analysis. It then gives you the opportunity to give more precise advice, to identify if you, or if you're working in cohorts, are susceptible to certain types of drugs or less susceptible, whether you're more inclined to react positively to a Mediterranean diet or a ketogenic diet, or to make the appropriate lifestyle adjustments that you need. But to make these technologies available, you have to drive the costs down. They have to be not only cheaper, but easier to use. And we still have a ways to go. With every precision health workflow, whether it's commercial ones or ones that are being conducted in laboratories around the world right now, there is sort of a process. There's sample collection, there's analysis using different instruments and different tools. In almost all cases, there's some kind of data analysis involving both databases, reference values, but also integrated machine learning to interpret these and then to produce outputs and results. Determine whether you are above normal, below normal, just fine, whether you're in the green zone, which is good, the yellow zone, which is a warning, or the red zone, which is bad. And that's how many reports for commercial companies and also for university-based research follow. Once you've got those outputs, it's not just simply a report, you need advice. And that advice is where the real difference is, and whether that advice follows changes in nutrition or supplements or drug therapy or exercise and other lifestyle adjustments. Those have to be personalized. They are that N of 1 concept because everyone is different. And the idea is if you can measure things unique to that individual, then you can prescribe solutions that are unique to that individual. These are examples of some precision health reports produced by a company called Nutriwell. This one measures about 720 different metabolites, and then it integrates that into a whole bunch of different reports covering about 35 different conditions. So this one identifies your risk for type 2 diabetes and insulin resistance. This particular individual has a medium risk, or some things that are out of range, particularly one important compound that's out of range. It produces an overview of all of the measures and a score. This person got a C instead of an A or an A+. And it provides very detailed reports about what's gone a little wrong, what's going a little right, and then also a very detailed prescription about what to do about mitigating the risk. So this person does not have type 2 diabetes, but they fall in a domain where they are potentially at risk. So if they were to get diabetes, perhaps it's 10 to 20 years away. The idea is an intervention now can help. So I'm supposed to talk, and some of the other presenters will also talk a little bit about how artificial intelligence and machine learning can help in this process. So the example I gave of the NutriWell report required analyzing 720 compounds quantitatively. So you had to do the whole metabolome analysis very quickly, very cheaply, and you want to be able to do it on a regular basis, on a monthly or biannual basis. So using AI to help with data analysis for MS, from AI peak picking, integration, calculation of concentrations, that's something that had to be developed and still is being developed. The other thing that's going on is that every day there are papers, dozens of papers being published about biomarkers, about changes in both metabolites and proteins that affect a person's risk. None of us can keep up with that. So using things like large language models and natural language processing allows you to automatically mine thousands of papers and dozens of databases to pick up on the information that's being added daily. So in the case of NutriWell, they use a lot of that infrastructure to mine what's going on a daily basis and to adjust both the reports and assessments on a regular basis. Same thing is possible to use large language models to interpret the data, also done by NutriWell and by other organizations and companies that I've mentioned. So they help interpret the data. They also have the analytical performances. I've highlighted how some of the things like large language model text mining and reasoning is possible. In these cases, we would have to use knowledge graphs, and other companies are beginning to use this. This avoids hallucinations and gives you references that allows direct citations. It also allows you to develop precise intervention and precise prevention plans that are well supported with evidence and references. These are some examples, again, taken from NutriWell, where they've got reference databases, reference values, biomarker patterns, and looking for specific metabolite trends and patterns to look at these specific conditions. In this case, you're seeing elevated levels of tyrosine, alpha-ketoglutarate, TMAO, which are in the red zone, and then others in the yellow zone. And this also indicates someone at risk, rather high risk, for metabolic syndrome. The diet-metabolite relationships, even drug-metabolite relationships, are also hard to track, but using AI and large language models helps identify those very rapidly, very consistently. As well, it allows you to put some recommendations that are both logical and consistent. Now, it can't be done entirely through AI, but when you're writing these things up, it does take a fair bit of time, and so AI helps with that. And so these are some of the recommendations that are partly gleaned from the literature, but then validated by registered dietitians. So I wanted to give just a couple of very short case studies where the idea of continuous and regular monitoring makes a difference. And this is a case or situations where in one case it wasn't one person, but it was 122 people. They had both their metabolomes and their proteomes measured, and they were given essentially a health score. This is from a company called Molecular U and was published a couple years ago. So the score ranges from 0, which is meaning you're dead, to 100, which means you're probably an Olympic athlete. And they were rated in terms of their dietary health, cognitive health, gastrointestinal, cardiovascular, liver health. And so they had initial starting points. People sort of self-selected. They generally thought they were healthy. Interestingly, most were unhealthy with scores below 50 or average scores below 50. Each of them was given an individualized plan where they had to both change their diet and their lifestyle, and they were tracked over a little over 3 months, 100 days. After the 100 days of that intervention, they were then tested again. And in every category of health, there is an improvement. In some cases, improvement was quite substantial. Obviously in the dietary health, but interestingly in the cognitive health and in kidney health. Liver and diabetes also improved, not as spectacularly, but across the board. So it wasn't a case that all 122 people were given the same recommendations. They were given 122 different recommendations. But the other thing that's surprising is that just in 3 months, you're able to see substantial improvements. So that's sort of a proactive plan. There's another one which is more passive, but in the sense of just regular monitoring. Not quite sure why that one went there. But this is something that we published a couple years ago, where again, this is an individual who had been monitoring her health through metabolomic and proteomic testing quantitatively for a number of years. He started, I think, in 2016, but just sort of showing where monitoring was happening of the relevant period. So in 2018, blue means perfectly fine. 2019, there's a little bit of red. 2020 was the COVID pandemic, so she went back to test in 2021, and you start seeing red appearing. 2022, it's essentially all red. And the trend at that time was, okay, this is alarming. She felt fine. What's going on? The AI said that she either had ovarian cancer or pancreatic cancer. And so she was asked to take an MRI. And the MRI revealed a growth at the tail of her pancreas. She had to go back multiple times before anyone took it seriously, but eventually she was able to get someone to do a biopsy of that growth at the tail, and she found that the CEA antigen levels, which are normally about 3 or 4 nanograms per mil, were 12,000. So they removed the tumor, and she has since been cured, and this is the first example that the surgeon or her doctor had ever seen of stage 1 pancreatic cancer being picked up. So this is how regular metabolome monitoring can save lives and also lead to precision medicine. So to wrap up, I think we wanted to contrast the differences between precision medicine, which is an interactive, often associated with medical interventions, versus precision health, which is a proactive approach to monitor and change your own health. So I think I'll wrap up, and we'll have our next presenters, and Susan is coming up.
We will take questions after the third speaker, and hopefully we'll have shorter sessions 4, 5, and 6. Thank you, David. Fantastic talk.
Great to see everybody, and I'll be talking about nutrition for precision health and try to link into what some of David was just talking and Rima had spoken about. Okay. So why nutrition matters? Well, it matters in metabolic individuality and health and disease. And we know that our essential nutrients are compounds that are supplied from our foods. We can't synthesize those in our body, or can't synthesize them in the amount needed. So we have to get them from the food because they're needed for normal growth and development and reproduction. Nutrients provide energy, and important to this group, they serve as cofactors in hundreds of biochemical reactions of host metabolism. So don't forget that when you're analyzing your metabolomics data. They serve as antioxidants, are involved in gene transcription, DNA methylation, and they provide structural constituents for the formation of cells, bones, teeth, and blood, to name a few. We do know that nutrition plays a role in mitigating the onset or progression of disease, but we don't know yet how to prescribe nutrition. We do know that the optimal foods or level of nutrients can depend on genetics, lifestyle factors, life stage, states of health and wellness, and many types of exposures. Exposures to components in the foods and beverages that could be good or bad for you, but also additives and impurities that the food industry intentionally puts in our foods that are endocrine disrupting compounds. Drugs and medications also can influence what our nutrients are doing, and other sources of environmentally relevant exposures and pollutants that we may get inadvertently. So I will be talking about nutrition in the context of a precision medicine initiative which is called the All of Us Research Program. It was initiated in 2015, about a decade ago, which aims to include over 1 million diverse participant partners. As of March 2026, there were 560,000 who have completed the baseline enrollment in All of Us, and with more than 870,000 that have enrolled. So this is a longitudinal study and captures multiple data types, electronic health records, surveys, baseline physical measures, biospecimens, and genomics, to name a few. The Nutrition for Precision Health study is an ancillary study or a subset of the All of Us program. The primary goals is to develop algorithms to predict individuals' responses to dietary patterns or dietary intake. This involves gathering a comprehensive set of microbiome, genomic, physiological, metabolic, behavioral, cognitive, contextual, electronic health records, surveys, and environmental data. A large and diverse population of participants are participating in the Nutrition for Precision Health study, and the primary purpose is to inform tailored interventions that improve health. The NPH study uses a modular approach. In Module 1, it's an observational study to examine the baseline diet, or some call the habitual diet of individuals, and physiological responses to what's called a meal challenge. There are about 8,000 participants in this Module 1. In Module 2 and Module 3, it's controlled feeding studies where individuals receive each of 3 Nutrition for Precision Health diets as a short-term intervention. Module 2 is in at-home or in community dwelling, about 1,200 participants, and Module 3 is live-in controlled feeding studies, about 150 participants. The program is structured with 14 clinical sites across the United States to ensure diversity in the population being recruited, but also geographic location tells us more about seasons and diets in those regions, and also their environmentally relevant exposures that can differ at these different sites. These sites do the recruitment, enrollment, the surveys, all those kind of things, and they send biospecimens to the Metabolomics and Clinical Assay Center that I direct, a Microbiome and Metagenomics Center, and Dietary Assessment Center. All this data comes together with GWA data provided by All of Us to do artificial intelligence, multimodal data modeling, and bioinformatics. Okay, so a little about these diets. The NPH diet has 3 diets that each person consumes in a crossover study: the blue, the purple, and the orange diet. These diets vary in the amounts of fruits and vegetables, whole grains, beans, meat, poultry, eggs, fish, nuts, seeds, vegetable oils, dairy products, and sugar-sweetened drinks and desserts. And so we can know more about how individuals respond to the diets by having those various components in each of these diets. Now, Module 1 participants complete what's called a liquid challenge for baseline evaluation of the response to what they typically eat, their habitual diet. Module 2 participants complete an actual meal challenge at the end of each 2-week diet, and Module 3 participants complete both the liquid challenge and the meal challenge. Blood is collected at the end of each 2-week diet before and after the challenge. There's like 9 time points that are collected for up to 4 hours. Metabolic phenotyping is conducted via clinical assays to determine the response curves for clinical analytes for insulin sensitivity, beta cell function and insulin response, and lipid metabolism. The results tell us how an individual responds to each diet in comparison to baseline and to the other diets. So the measurements are these test diet interventions, the liquid meal and diet-specific meal tolerance test, our questionnaires, dietary assessments, and lots of physical measures. Now, in addition to the clinical assays, phytochemicals are also being measured, and metabolomics is being conducted using these blood-derived samples, these phytochemicals, to really sort of help us understand what are these good things in the food that are important to understanding people's response to diet. Saliva collected for GWAS and metabolomics. There is homogenized food saved for composition, phytochemicals, environmental analytes, stool for microbiome, metagenomics, untargeted metabolomics, and urine for determining energy metabolism from double-labeled water experiments and metabolomics, and then hair and nails for environmental metabolomics. Okay, so what do we do with this data? So what happens is all of this data comes together, the microbiome, the metabolomics, the GWAS data, the clinical data like a comprehensive metabolic profile that one would normally get or traditionally get at an annual wellness checkup, continuous glucose monitoring, accelerometry, all those questionnaires, and then results associated with these meal tests. Now, to use the term that Di Thomas at the AI Center uses, this undergoes data wrangling for feature engineering and selection, and the result is to determine those features that predict the diet-specific meal test response of the area under the curve of those clinical assays, which are so easy to measure. Okay. Now, the center that I direct is that Metabolomics and Clinical Assay Center. I told you about the actual clinical assays used for the developing the area under the curves, and the quantitative targeted phytochemical assays to look at how do these good phytochemicals in our foods predict individuals' response to dietary intake. We're also doing quantitative targeted assays of host metabolism like acylcarnitines, amino acids, biogenic amines, branched-chain keto acids, and untargeted metabolomics of what I call the internal exposome using high-resolution MS. So since this is a mass spec audience or a metabolomics audience, I'll tell you a little bit more about what's being measured and why on our untargeted metabolomics platform. We're looking at the simultaneous measures of these analytes. And we've acquired a spectral library of over 5,000 compounds at this time so that we know the range of analytes that we detect on our platform. And of course, we also use public database matches in addition to our retention time exact mass MS library of these 5,000 compounds. This covers host and microbial metabolism of over, say, 1,200 compounds of amino acids, polyamines, bases, nucleosides, and tides, carnitines, sugars, disaccharides, fatty acids, lipids, steroids, bile acids, hormones, to name a few. In addition, we do have a subset library that's simultaneously detected of compounds that are related to diets that are rich in good polyphenolic diets. And so that's what we call our food metabolome. Simultaneous to this, we're detecting many environmentally relevant metabolites. In fact, we acquired data for what's called the EPA ToxCast library of over 4,000 compounds that are priority chemicals of concern, many of them being endocrine disrupting compounds, some of them being intentionally added to our foods in the United States by the food industry. Some of them, though, we're being exposed to because of the agricultural, livestock, and poultry use, like antibiotics, pesticides, insecticides, and herbicides, pollutants like PAHs, smoke. Some we're being exposed to from the home, garden, and personal products, like all of those sides I just mentioned, the pesticides, et cetera. Parabens, phthalates, flame retardants, food additives like the parabens, the BHA and the BHT. And remember, BHA is removed from foods in Europe, has been banned for quite some time. Packaging foods and beverages, phthalates, as you know, are in a lot of those. They have a lot of health consequences. And then there's many food contaminants. So when we look at environmentally relevant analytes, let's not just think about, oh, we're walking around and we breathe them in, but think about the fact that they either are in our foods because somebody put them in there intentionally, or they've gotten into our foods inadvertently. Okay, last I wanted to mention is that understanding what's going on with those essential nutrients simultaneous to all these other compounds is important. So we do have a good understanding of one-carbon metabolism, like choline and folate metabolism, our essential amino acids, carnitines, acylcarnitines, the omegas, and many, many vitamins and vitamin-like compounds. It's really important to be looking at these as we look at our environmental exposures and our host metabolism, because it's known that environmental chemicals, drugs, and medications can interfere with the transport, metabolism, or utilization of essential nutrients and perturb endogenous metabolism. And so I encourage everybody that's doing metabolomics to make sure you look at what's going on with those essential nutrients, because this will be the way that we can derive nutrient cocktails that can mitigate the effects of environmentally relevant exposures. Okay, I'll end there. Thank you.
Hi, is it on? Yeah. Question for David. So you mentioned that ideally these quantitative, commercial or not, assays should measure at least 500 compounds. And I'm wondering where you got that number. Is that a specific targeted list, or is it a statistically derived list that could be targeted or untargeted?
Sure. So go up to the mic here. Yeah, it's sort of a consensus, and obviously you could have 500 obscure compounds, and it doesn't relate to it. But if you're trying to capture a good portion of the major pathways, it kind of comes up to about 500 or so. But the extent of coverage that people might want, obviously more than that. But this is sort of a minimum set. Yeah.
Hi, thank you for the talks, very interesting. I'm curious about the reference values that were represented. So how those references values for metabolites are defined, because I don't know which population, men, female, there is differences and there's just a range, so I don't know if we can have more information about that.
So is she asking about sex differences in metabolism?
It was a little hard to see here.
Were you asking about sex differences in metabolism? No. I wasn't. I heard
No,
male and female.
I don't know. Yeah, I was talking about the reference ranges that are defined, who is defined them and how are they defined? Which population is studied to make those references?
Yeah, I think that's a very good point. There are quite a number of formal reference ranges that are published and are in both public databases and in books. And those have been collected both for male and female and for different age categories. And they are quantitative, so these are derived from clinical measurements. So there's the Caliper study is one that was done in Canada. There's been others that are published, as I say, in some commercial and in textbooks. We've integrated a lot of those reference ranges into a database called MarkerDB. And so that covers the different ages and sexes.
Yeah, I wanted to add to that and say that I'm very interested in reference ranges also, because those reference ranges, like for clinical assays, for example, are just arbitrary. I mean, we didn't really know what they are, and nobody really knows what they are now, and that's beginning to, come out more. I mean, we know what's reported, right? But we don't know exactly what those reference ranges should be, because they were really established very early on using primarily, I think, Caucasian male reference values. And so now, what does that mean if you're not a Caucasian male? So we're beginning to understand more about that. One thing I did want to mention is that in a lot of my studies, the clinical assays on individuals will be within the reference ranges based on their sex, for example, but we will see big differences in our, say, OPLSDA models when we're looking at like case controls in our metabolomics data. So just because the reference ranges are within normal really doesn't mean much. And, I think that we're going to really be able to move the field forward in understanding exactly what the medical doctor should be measuring, because we know those comprehensive metabolic panels of those entire 14 analytes that you get at your annual visit when you get to talk to your doctor for 2 minutes aren't doing what we need them to do.
More data on populations is needed, whether Asia, Europe, we need more. I think it's a hugely important question. Any others? Okay.
Yeah, so I was curious about different timescales that you were choosing for different analyses for all of the discussions. So for molecular U, how often is that sort of thing done? And I guess, is there some sort of relationship or balance you can strike between, okay, we'll maybe do a dozen analytes every single day, or we'll do 500 analytes every single month, or something like that? What are your thoughts on these ideas?
So the monitoring with molecular U is typically done annually, and that seems to have been in sort of a sweet spot. They're primarily looking at chronic conditions. So, an infectious disease, they're not going to pick up. But if you're trying to look at cardiovascular disease, Alzheimer's, Parkinson's, cancers, those generally take years to develop, and so that's where they're generally targeting. I think if you're looking at infectious disease, or if you're looking at response to diet, then almost weekly or even monthly. Mike Snyder, many of you might know, does it hourly or every few hours. So, that's possible. But I think in terms of, what's reasonable for a human, once a day seems to be sort of the max, and we've done some tests with that.
So I think we need to do better. 1 year is not enough. What's the point? I go to my annual checkup once a year. I need to know in real time, meaning all the time. Yes, I have my watch, I have my, I need to also know my metabolic profile. We need to do better. We need to know it in real time all the time. Please watch out for a proactive health initiative, a public-private partnership that we are going to announce soon to benefit the public and by our community. Please join our precision health, precision medicine working groups within the Metabolomics Society and the MANA, and wherever you are globally, let's do better. Let's bring the bar higher. Let's hear from you. Yes, let's do better. I just want to allow the last 2-3 speakers to comment. Yes, one last.
Thank you. So going back to the discussion about reference ranges, I was curious about what would it take, because this seems like a pretty pressing issue from what I can hear. What would it take exactly to get an accurate characterization of the different reference ranges for different populations, or even for those N of 1 patients, for every person that you're testing?
Yeah, and this is maybe where Susan and I perhaps disagree. I've spent most of my life trying to get reference values. That was the reason for the Human Metabolome Project, the Human Metabolome Database, development of the databases and marker databases. So I've spent 25 years trying to get quantitative reference values for hundreds, if not thousands, of metabolites. Those have been determined. They are now available. They are useful. And it is a lot of work. But my my whole essence of my work is to make that information public and to provide that to the community. So we have some very good reference ranges now that are for healthy individuals. What they are for specific conditions is where the rest of you need to work on. Some of that is starting to appear now. I mean, we know the reference value to define diabetes, 7 millimolar glucose. But there are other reference values that are starting to appear for certain conditions. And that's where this marker DB is, I think, useful. And again, it's publicly available. But it's only partly complete. And I think this is where we need the community to provide these reference values and reference combinations, because it's usually not just one metabolite that defines a condition. It's also where AI is important. It's where this idea of integrating all the information that's being published in thousands of papers literally every day so that we can stay on top of that.
Very good. Thank you. Thank you, David. Thank you, Susan.
Okay.
Quick remarks now.
Very good.
From the last three speakers, Matej.
Okay, excellent. So thank you. So I will move on. So I will want to highlight basically what already David was highlighting, and that is the point being that disease process is complicated, and there are many, many events happening already before there is a threshold for clinical diagnosis. This is important to understand these underlying processes because that will help us detect disease early, or at least risk of the disease. And ultimately, if we understand these underlying processes, and this is something that we don't have really too many success stories there so far in any of the diseases, but there is potential for early treatment. There are some diseases that are preventable, like type 2 diabetes and steatotic liver disease, so there definitely early detection would be very important. And one has to keep in mind also that this is simplified graph, conceptual graph, because, for example, if you have a glucose that is elevated, that by itself is not going to be a problem, but there is a path towards many possible comorbidities, and that is exactly why the diabetes is so harmful. So you have cardiovascular disease, you have kidney disease, you have various kinds of other complications. So this is what we try to prevent. Ultimately, what you want to study then, to be able to study this sort of progression of disease, you need to have a longitudinal data. You have to go very early, from early life, and what I will try to focus on, show you, is basically the early life events in some diseases and some examples. So first, type 1 diabetes is one example where there is definitely a lifelong process. What do we see? Yes. So, and this is just an example of one girl who developed type 1 diabetes at the age of 9 years, and this is the profile of autoantibodies. So keep in mind that you should develop these antibodies already 2-3 years ago. So if you have 2 antibodies or more, it's almost certain that you will get the type 1 diabetes eventually, but we don't know when. And these are not symptomatic, so antibodies will not tell you. If you go to the clinic, nobody could tell you that something is wrong. And routinely, we don't measure antibodies in the clinics. So this is a typical example of importance to be able to understand some of these early processes. So we have done actually quite a long time ago, we worked with a type 1 diabetes cohort, and what we found, for example, that if you have this longitudinal series, and there is a very large 8,000 children cohort who are all at moderate to high risk of HLA, HLA-associated risk, so they, and then you need to have a stratification based on genetics, otherwise the prevalence incidence is not high enough to get enough power, even with 8,000 children. So, but we found systematically the lipids were lowered in these children who later on developed type 1 diabetes. And that happened already before the autoantibodies appeared. So there is something happening already, possibly prenatally, that is causing this disease to happen. So we have found that, therefore, that it's in utero metabolic risk, but what could be the cause? And there is some interesting data showing in Finland and Sweden, for example, that since like 2000 or so, a little later, so last 20 years, the actual incidence has stabilized. So it has been rising since 1950s almost, and now seems to be flattening. the reason what we found was because obesity cannot, viruses cannot explain this microbiome diet, they cannot explain this pattern change, but there is potential for the, for example, environmental chemicals such as PFAS, who have started to be more regulated in Europe in about 20 years ago, particularly PFAS was banned. And this is just an example. We measured in a mother-child cohort about 264 mothers and infants. We measured in the mothers PFAS, and we found that actually just measuring PFAS and developing a model, one could reasonably well predict the future progression to autoimmunity. So those children who have progressed to islet autoimmunity in the follow-up, like 3 years of age, they actually had a specific profile, maternal profile, measured using the PFAS alone. I will skip the bile acids. Just want to measure that we have ongoing project initialized that we are leading, where we are looking at several birth cohort studies, which are very well phenotyped and having different kinds of outcomes. Obesity one, then there is also autoimmune diseases, allergies, but then also neurocognitive outcomes. And we have a number of functional studies ongoing within this, also within this project to validate the findings conceptually. So for example, there was one study we did looking at, this is in the Swedish ABIS cohort, where we linked the at birth and age 1 profiles. We linked them with the future neurocognitive and neurodevelopmental disorders, including autism. And another study, we also looked at, this is the functional outcomes. So we look at the prenatal exposure, maternal exposure to PFAS, and actually being able to link them to the MRI image outcomes, adverse functional outcomes in children who were 5 years of age. So that's what MRI imaging was done in the babies, children, young children. So it's certainly possible to look at the exposures and also metabolome in early age and early life, and then link them. But you do need to have a longitudinal prospective data, and that is the crucial part to be able to predict the early stages of the disease. So with that, I would thank to my colleagues and collaborators, and of course the funders for
Tuulia.
Okay, and now we have been talking a lot about the biomarkers and predictive patterns. So now I'm going to talk a little bit that it's not enough just to have biomarkers. So we are good at finding them. A lot of these have been reported in the literature, but actually not too many have been taken to the like a clinical use yet, and that is because it's difficult to validate these markers. So because if you have these biomarkers like disease biomarkers, exposure biomarkers, predictive signatures, multi-genomic classifiers, we have to know what is the biological origin, which is the tissue, or maybe it's gut microbiota that's producing them, what are the metabolic pathways that drive them, and are they causal or reactive. Basically, it's also important to understand that if you find a biomarker, it doesn't really tell you too much about the mechanism. For example, if you take hippuric acid, it has been used often a marker of better metabolic health, marker of frailty in elderly adults, so lower levels are markers of increased frailty. It's also a marker of greater microbiome diversity, and because it's derived from the food, so also higher fruit and veggie consumption. But can we conclude, does the hippuric acid improve the health? And just a very quick wrap up. So basically, hippuric acid is produced by the gut microbiota from the dietary polyphenols. They form benzoic acid, which is conjugated with glycine in the liver, and then you have hippuric acid, which is part of it stays in the circulation, large part is excreted via urine. But what could be leading to the higher levels of hippuric acid and why it's beneficial? So basically, it's higher intake of plant foods, of course, that is beneficial. You could have a more diverse microbiota, or you could have a more efficient micropolyphenol metabolism, or you could have differences in your hepatic conjugation, or it could be excreted a little bit slower. And metabolomics alone cannot distinguish which the mechanism is. So you need kind of a different type of model systems. So in vitro, you could do fecal fermentation, study on polyphenol metabolism, try to identify which are the microbes producing the benzoic acid. You could use animal models, germ mice, antibiotic mice, so that is changing the gut microbiota, dietary intervention studies, and of course human studies, but like controlled feeding studies, organ-specific sampling, longitudinal intervention studies. So you need a multiple level of studies to understand where it is coming from. And it's really, as said, important to understand where the metabolite is produced or where it's obtained. So is it the host-related metabolome, microbiota-related metabolome? Is it coming from diet, environmental exposure, or combination of these? And where is it transferred? Gut, liver, kidney, and the pathways that regulate it. If you don't have this information, so you can't really interpret where the biomarker is coming, and with that you get also Bruce translation of the biomarker in a treatment possibilities. So basically, this kind of functional validation methods usually use in vitro models, different kinds of cellular systems, from simple systems to organoids, or even like gut-liver systems, or advanced ones. Ex vivo systems, or for example, liver slices, a lot of animal models, and human intervention studies. And it's important that no single method answers to all questions, so you need different levels of information, and you get one piece of puzzle and maybe get a more clear picture when you combine all of this. So I'm showing a little bit on our approach. So basically we are looking how the environmental exposures, chemical exposures are having an impact on the muscle progression, so metabolic associated steatotic liver disease. So we have started this already like 15 years ago, looking actual what kind of metabolic changes we see in the muscle patients, in the liver, in the circulation, a lot of different types of studies, liver biopsies, dietary studies, looking where the lipids, for example, which you see, which lipoprotein fraction they are. So very comprehensive studies. And then we added up the exposome here. This is part of the EU project I'm coordinating together with Matej. So we have been looking plasma samples from the European SLA registry, looking metabolome comprehensively, looking in vital exposures. We have for 2 different cohorts of liver biopsies and matching plasma samples to see the organ level changes. We have a catheterization study where we can see fluxes through the liver, so what is going into the liver and out of the liver. And this is a very tedious operation, so that is a small study. We combine that with the mice studies exposed to PFAS now when we are looking, where we have blood, liver, intestine species, and actually also urine. Then we have different hepatocyte models, like PFAS exposure with or without steatotic phenotype on the liver cells. We also do more targeted ones, so we do combined omics, transcriptomics, also in the cell line studies, this phenotypic high-content imaging. And we also isolate, for example, mitochondria, which we have been seeing is specifically affected, so we can see like detailed information on the level of the cells. I'm not going to tell too much of that. If you're more interested, I have a talk tomorrow, and my postdoc show has a presentation on Wednesday. Basically, key findings that we could see that PFAS exposure is exacerbating the metabolic dysfunction in muscle patients. It's sex-specific, especially in lipid metabolism, but also a lot of these changes are related to gut microbiota and inflammatory signaling. And we see consistent findings across human, mice, and hepatocytes models, but they are not fully overlapping, but we get complementary information also on the model studies. And of course, then we see a lot of associations, and it's important to see what is going from the association to causality. So what is actually the key event that is happening, and if there's any confounding factors. And here we can use different types of studies to see this causality, like G counts, microbiota manipulation, so germ-free-wise, antibiotic treatment, and so on, or even changing of the gut microbiota, different kind of stable isotopes tracing, like flux studies, and targeted interventions. And it's important to understand that these causal pathways are much more valuable than the biomarkers alone, because then you can understand what is the mechanism behind the actual biomarker. So just summarizing, so basically biomarkers alone are not sufficient. We have to understand what are the biological sources. We need functional studies for interpretation, where they are coming from, what are the pathways, integrating different level of information, integrating not only metabolomics but also other omics levels, and understanding the pathways transforming biomarkers into the actionable knowledge. And the idea is, of course, we don't only want to identify who's at the risk, but why the person is at risk, because then you can start developing targeted treatments. Okay, thank you.
Okay, Jennifer. We do want to leave a few minutes to interact and get feedback and to connect with the coordinators of the first session that we attended on big data and biorepositories. I see many of the leaders are sitting in here. You are dealing with big data, biorepositories, and all complexities. We are dealing with big data to understand individuals and variations among individuals. Let's have a dialogue. Where can these meet?
Okay. Buenos dias. That's about the sum total of my Spanish, but I'm really pleased to be here. I've been asked to keep this very short so we have time for discussion. This is a talk on frustration. And for once, it's not frustration with Buenos Aires traffic. It's personal frustration, because we have been trying to translate all of these type of findings into differences in people's lives. So this takes good communication, but mostly it takes money, or at least you have to think about the money, how you're going to fund it. But I think what is often left out is what's the benefit for the funders themselves. And there's an optimistic side here that I hope as a community we can change the things that we cannot accept, but also recognize the things we cannot change. Whether I have the wisdom to know the difference, I'm not sure, but I hope you do. And apologies to Reinhold Niebuhr for taking his prayer and using it for this purpose. So keeping things very short, why do we want to translate? Because this is where we have real-world impact. And it really puts together the biomarkers that we have discovered. I totally agree with Tuulia that we need to think about mechanisms, and we also need to think about this so-called grey market, where you have direct-to-consumer tests, some of which are better validated and have better underpinnings than others. And I'd like to acknowledge the people in the field who are really working hard to make sure that these direct-to-consumer tests do have a scientific basis and a biological basis behind them. And I think the worst reason to translate something is for your own financial benefit. But we cannot ignore that hopefully there will be a financial benefit to somebody. So I'm going to be quick, but these slides will be available. The average time to translate takes between 15 and 20 years. Now I want you to think about this for a moment. 15 years. Our patients don't have 15 years to wait. Neither do we have the time and funding to have such a high attrition rate in this so-called valley of death. So we can do a lot with precision medicine. One of the challenges that precision medicine gives us is it subsets groups. And this gives us higher bars to reach statistically, but it also offers an opportunity that possibly we can better choose cohorts to take forward. So we have risks and opportunities, but mostly we want to fail often and fail fast in the process to give us better translational opportunities. We can use AI to our benefit to make sure that we can get faster throughput on this and maximize the interpretation. And I'd like you all to think about AI as a companion, not as a replacement for yourself. You are the best tool that we have. And I know that Rima wants me to finish, so we have time for discussion. I am going to say don't neglect the admin and technical support, because what we really need is to have a whole translational ecosystem for this to work well, with sustained funding across the pipeline. And most of all, and I can't discuss this today, but we want to make sure that our results are interpretable, particularly when we're using AI, because if they're not interpretable, we have the risk of, let's say, unintuitive decisions that could affect patient safety. So I'd like to finish with the success stories, and I'd particularly like to highlight that untargeted metabolomics is beginning to move into the clinic. It's really doing great things, and I'm going to go over to the discussion.
Gordon Conference, Jennifer, Gordon Conference.
Yes, and thank you, Rima. So we have a Gordon Conference on Human Health and Metabolomics starting on the 28th of February. There's a slide that is actually not on this group of talks, but anyone who's interested, just email me or register direct. It will be great. Rima will be there. And we hope that all of you will be too.
Great. And here is Matej, who was the co-founder of this series. Matej, it was a great pleasure coordinating this with you. Please, the first session members are here. Big data, you have witnessed some of the things we are worrying about as a community in thinking about human disease, variations in trajectory of disease, leveraging big data that is out there. Yes, some of the lessons you have learned, some of the lessons we are learning in data harmonization. And when we talk about data harmonization, we are trying to bring the lipidome, the metabolome, the exposome, try to connect to derive knowledge, a different perspective on what you call data harmonization. Any comments you want to make?
Okay, so I think that let's start. So when one is combining different types of omics data together, like what you are trying to do here, I mean, there are so many ways to do it that that is essential to have transparency in how this is done. So, and ideally, transparency is the first step. So to understand how other or your colleagues have done it, and the second step is to try to do things in the same way, and that's more kind of towards standardization. And in order to enable that, then the data needs to be available somewhere, and it needs to be available to the community for whatever mechanism that it can be made available. So I don't know. These are my first thoughts. We can continue the discussion.
Yeah, we'll continue. Any other comments? No. But when we talk harmonization, we are just harmonizing metabolomics data, right? We haven't even discussed the rest across omics. Comments, remarks on what you have heard today, what would you like to see? White paper. We are working on a white paper on the concepts. All are invited. Please reach out.
You want comments? Okay. I want to echo what David said. In order to make metabolomics to be relevant to clinic, one important thing is quantification. Just because when you go to clinic, doctor ask a number. They ask a number. They don't care how you measure it, but they want the number. So the quantification is very important, one thing. The second thing is when we see like big data you're talking about, like all of us, all those, one thing that's very important we need to look into is the patient follow-up. Because if you have the data, you have the sample you collected 10 years ago, and by now you have no idea what patient is doing, no use. So you have to be very careful.
So I can tell you in brain research and in Alzheimer's, we are going back 20 years back, 30 years back, and gathering all information forward to see what has happened. That's what you need to do, absolutely.
But with the big data bank, you have to
exactly. So from disease to biobanks to big epidemiological studies. Okay.
I just wanted to comment on the question of microphone.
Should sit better. Well, it's a good idea. If we all sit up there, yeah, it's make it easier. Yeah.
So we've just heard twice today that quantification is important. And I want to emphasize that I actually agree with that before I make my next point, which is that it's not necessarily essential. We've traditionally seen it as essential, mainly because regulatory bodies have insisted on it, and it makes our lives easier. But there is one regulatory test that does not require quantification, and that's histopathology. It uses pattern recognition. And I think that we are reaching the stage technologically where it may possibly be relevant to consider untargeted metabolomics as pattern recognition, and as long as you can show that it is reproducible and stable, it may be possible to use it as a diagnostic tool without absolute quantification.
Questions, comments?
Yeah, I was just going to say I agree on the point there. In particular with personalized metabolomics, if you build a longitudinal model of someone's metabolome, and this is a relative model of their own variations internally, but you don't actually have an absolute measure, you can still gain a lot of value from that data that we've seen ourselves can be very valuable, even with small groups of metabolites, because the interactions between metabolite A, metabolite B, metabolite C, and how they move over time can give you a lot of insight. So yeah, essential is a word we should be very careful with when we're trying to make things cheaper and bring them into people's lives.
Within an individual, I think, right on. Other comments?
I'm just curious about one thing, that if you're comparing two groups, one is a control group and like healthy control, and another group where of a particular disease where the person has become drug responsive and the symptoms and the overall disease has subsided after a period of time. So the metabolic profile of the two groups, like essentially, will they overlap? And if they don't, if that is a contradiction?
Any responses? Quick, please, responses.
Get that to come on. Oh, there you go. High technology here. You had to find the right button. Okay, so I think most of the designs, maybe you have the case versus control, but then the responders versus non-responders within the case group. And so that's how I would look at that. The other thing is more studies that I'm doing now are case versus cohort rather than case versus control when it comes to larger epi studies. And that gives us a real opportunity to look at that across that entire cohort and instead of at just a particular selected control group of individuals. I think that makes the study much stronger that way, and is something that I've moved to in my own studies.
What would you like to see the Metabolomics Society do, the community do? We have created vision, we have moved a long way for 20 years. Where should we go?
Yeah, I'm not sure if it's exactly on that point, but might foster the discussion as well. And yeah, I don't know, it's probably more related to what David, Matej, and Tuulia presented. But I would like to hear maybe what's your opinion on the role of the exposome on this, because when I think about all these metabolic changes, we now know that these environmental stressors as you also showed, play an important role. And when we see these metabolic changes, it might be already a consequence of these exposures. And when I think about these personalized kits, for example, how would you see us implementing, for example, tracking the exposome and these environmental compounds? And maybe a follow-up kind of question based on the other questions. Then for that, we probably need quantitative data, I guess, because we have regulations. So what is your take on that?
Before we get responses, just to let everyone know, there has been a phenomenal exposome summit in Europe, in Spain. Fantastic. The next one is in the US. Big initiatives funded under the exposome. Understanding and measuring the exposome and its link to metabolism is part of what we have as an initiative under NIH and under NIA in particular. Back responses from the speakers.
Yeah, so I wanted to start just by saying, yeah, I think exposome research is really critical, and that's where most of my work in metabolomics has moved to, of course. And these untargeted platforms will pick up many, many environmental analytes in addition to the host microbial metabolism and phytochemicals and essential nutrients. But we know it's really important because we don't have our endogenous metabolism perturbing so much just because we're aging. I mean, it does change with age, right? So guaranteed it does, right? But it's these endocrine disruptors that are in our environment and being put into our foods, which is probably what's causing a lot of the links to the diseases, right? And causes those metabolic perturbations. So they are really important to follow, but also we need to be following other things like stress, because we know that perturbs our endogenous metabolism. Also, family structure, how much physical activity one gets during a day. There are large metabolic perturbations that are associated with physical activity. So really looking at this exposome, not just the chemicals that we can detect on these mass spec platforms, but all of these other things related to, did you have a good morning? Or, you know, what are these stress exposures in your life and those kind of things. And to build the models that integrate all of that together is very important. And I do like to emphasize the diet, because really in exposome research early on, I think people were looking at, you know, if you go outside and you breathe something in, it's what you inhale. But really, our biggest source of exposure every day is what we intentionally put into our mouths.
Definitely. Matej, quick response.
Yeah. So first about that actually time course is very important, both chemical and other exposures, because sometimes actually exposures early can cause something much later. So that's one thing. The other about comment about quantitative. I think that for chemical exposures, it is important to at least combine untargeted and targeted, because for many, like PFAS, you are required from a regulatory perspective to actually have the quantitative measurements of PFAS. So it is important to have quantitative data as well.
Definitely. But I just wanted to put an alert as a community, a metabolomics community, right? And the people who are emerging as the exposomics community, the foodomics, we are all in part one community. So we have to be careful, right? We are measuring small molecules, and we see a lot of things in whatever, urine, blood, whatever. So I think we need to think carefully about building communities that bring these interactions all together, whether we are foodomics, drugs, drug metabolites, gut metabolites, chemical exposures. I just want to make sure we continue to build the interactions. David.
Yes, just to comment about the importance of the exposome, and I think you guys have heard a fair bit about it. We've been doing some calculations over the last number of years and have published a bit of this and still about to publish something else. But when you look at the fundamental or basic cause of death, which is different than what you see with the reported one when someone died of a heart attack, the question is, well, what caused the heart attack? And okay, if that was atherosclerosis, what caused the atherosclerosis? And if you go to the true cause of conditions, at least for the data in the United States, which is probably very similar around most of the developed world, somewhere between 90 and 93% of all deaths can be related to the exposome. You can do the reverse calculation, which is how many conditions or diseases are due to inborn errors of metabolism or genetically caused forms of cancer, which are the ones that you're born with, BRCA1, BRCA2, ApoE4 for Alzheimer's and other things. So if you do that tally, you come up with between 6 to 9% of all deaths are due to these inborn genetic conditions. So from two convergent approaches, you come up with sort of a similar solution, which is somewhere between 90 and 95% of your ultimate cause of death is the exposome.
And what we call big investments are currently available for modifiable factors of disease. That is key. If 40% of Alzheimer's is modifiable, why on earth have we not found at least agents that can slow disease progression, right? And we are doing this with the big data we are generating, but modifiable factors and the exposome is a big topic. Final concluding remarks, suggestions, white papers, engagement. We want to see young people lead and move us to the next phase.
So I think that most of these issue, especially then to make data available for AI, is very well connected to the previous workshop in the way that we spoke about data reuse and reuse of data in public repository, which is a huge treasure right now. And that's the reason why, and so this morning I asked the question about the issue about the clinical data associated with the data repository. So that is still a huge field of work, in my opinion, to go to that direction, in my opinion. So, yeah.
I think we need to have a voice. We've learned our lessons over 20 years. If we have a voice and if we are unified, we can have an impact. We can get invited around the table and we can motivate investments of funds, as we have done repeatedly and, within the NIH campus. So I think we have to be proactive. I think the barriers that are placed now in enabling US and Europe connect and the rest of the world connect, I hope these are not real. I hope we can overcome them, yes, and we do need a lot of money to reuse the data and make it available. Otherwise, what's the point? We spend so much money generating data. If it is not used and if our data is not replicated by others and if our data are not used by others who have different tools to find more interesting things than what we have found, leads us to the idea that an H index is no more, really is not sufficient. It's not. In today, the incentive must be an impact on moving a field forward where us and everyone else who used our data can get a credit. Any final, I'm happy to see a lot of people remained in the room, engaged. Anything? If not, I thank you very much and look forward to further discussions throughout the next.